With a show of hands, how many of us know someone who has suffered a stroke? Now, if you think about that person, consider what happened afterwards. How did they change, both in the short and long term? Stroke can cause debilitating impairments in memory, movement, and speech. Additionally, stroke is the leading cause for serious long-term disability with limited treatment options beyond stroke rehabilitation. While rehab is an important part of treatment, it is often very difficult for patients to fully recover. Thus, there's a crucial need for additional therapies towards enhancing brain repair after stroke. Stroke occurs when blood flow in the brain is disrupted and parts of the brain die. Most research in stroke focuses on fixing brain cells called neurons, but neurons are actually only half the cells in the brain. The other half are called glia cells, which are largely understudied. In my research, I focus on a particular type of glia cell, oligodendrocytes. These cells are really important because they help protect and insulate neurons through a process called myelination. Myelination occurs when oligodendrocytes create myelin, which wraps around neurons. You can think of myelin like insulation around electrical wires. Electrical signals are sent quickly along neurons, and this is possible because of myelin. This is how fast and efficient communication occurs in your brain. Stroke damages both oligodendrocytes and myelin. Without myelin and the oligodendrocytes that create myelin, brain signals slow down, which can cause impairments that we see in stroke. Despite oligodendrocytes dying after stroke, I found that in my research, precursor cells actually increase. Normally, these cells mature into oligodendrocytes, but instead, their gene expression is altered, causing them to be stuck in this immature stage, unable to move past this block and mature. In my research, I focused on stimulating these affected genes to facilitate repair after a stroke. I developed gene therapy tools specifically designed to modulate genes within oligodendrocyte precursor cells. Notably, targeting this cell type for gene therapy has not been done before. By implementing this therapy, we can restore the gene expression and remove this inhibitory block. The precursor cells can then mature into oligodendrocytes and restore lost myelin. By targeting oligodendrocyte precursor cells and removing this inhibitory block, we can repair the brain after stroke. This approach opens the door for new therapies for stroke patients to restore their movement, their memory, their speech, ultimately helping to achieve a full and complete recovery for our loved ones after they've suffered a stroke. Thank you.